Hello, everyone. Let's continue our class, pharmacology. In our first class, I have told you we divide pharmacology into 7 parts. The last part, the seventh part, is antibiotics. So, from this class, we will study the last part of pharmacology, antibiotics. This class, we will study some professional terms, and we should know these professional terms, conceptions. And then, next class, we will study different antibiotics. So, let's begin our class. Antimicrobial agents, general consideration. So, introduction. The last part of pharmacology, we will study antimicrobial agents, includes 3 kinds. The first kind, antibacterial agents. The second kind, antiviral drugs. And the third kind, antifungal agents. So, in your microbial class, you have studied about bacterial, and we used the Gram stain divide the bacterial into 2 kinds, 2 broadly kinds. The first kind, gram-positive bacterial, and retention of crystal violet iodine stream. The cell wall difference affecting antibiotic penetration, so stained into blue. What we call this bacterial bacillus or cortex. We call them gram-positive bacterial. And compared to gram-positive, the second kind, broadly kind, the bacterial be stained into red color. So we call this bacterial, they are gram-negative bacterial. We should study some professional terms, the conceptions. The first one, later we will study antimicrobial spectrum, and then bacteriostatic and bactericidal drugs. And then we will study chemotherapy index, CIOTI. The fifth conception is MIC and MBC. MIC, the full name is minimal inhibitory concentration. The last one is PAE, post-antibiotic effect. Okay, the first conception is the bacterial spectrum. Bacterial spectrum, the first one is narrow spectrum. Narrow spectrum means the antibiotic only against a single or limited group of microorganisms. And we call these bacterials, they are narrow spectrum antibacterial agents, such as isoniazid. Isoniazid, it is a drug, only can kill mycobacterial, So we use isoniazid only used for treatment of tuberculosis. And isoniazid is a narrow spectrum antibacterial drug. So that's now we have known the conception of narrow spectrum. The drugs means only against a single or limited narrow group of microorganisms, such as isoniazid, such as penicillin G. Penicillin G only can kill gram-positive bacteria, so limited group of microorganisms. Penicillin G is the narrow spectrum bacterial agents. The second kind, extended spectrum antibacterial drugs. These drugs can against gram-positive organisms and also against a number of gram-negative bacteria, such as ampicillin, such as amoxicillin, and these drugs, they are extended spectrum compared to narrow spectrum, such as this one, ampicillin, amoxicillin. We also call them broadly spectrum penicillin. Penicillin G is the narrow spectrum, but amoxicillin or ampicillin compared to penicillin G extended spectrum, so can against a number of gram-negative. They are extended spectrum antibacterial agents. The last cat broadly. Broad spectrum affect a wide variety of microbial species and are referred to as broad-spectrum antibiotics, such as later we will study tetracyclines, chromycin, and we will study quinolones. These drugs, they are broad-spectrum, means can affect a wide variety of microbial species. And broad spectrum means not only against the gram-positive, but also against gram-negative, and but also against some special bacterials. They are broad spectrum. So from this picture, we can know the first definition is the antibacterial spectrum divided into narrow spectrum, extended spectrum, and a broad spectrum. Okay. The second definition is bacteriostatic agents. What's the meaning of bacteriostatic drugs? That means these drugs only arrest the growth or replication of the microorganism. Means only inhibit the growth, couldn't kill, such as tetracyclines, such as erythromycins, macrolidins, and chromycin. These drugs only inhibit the growth of microorganism, couldn't kill bacteria, so we call them bacteriostatic drugs. Compared to bacteriostatic drugs, the next one is bactericidal drugs. Very easy, can kill the bacteria. We call these drugs, they are bactericidal drugs, such as penicillins, such as cephalosporins, such as quinolones. These drugs not only inhibit but also kill the microorganism. We call them bactericidal drugs. Number 4. The fourth definition is CIOTI, chemotherapeutic index. And in fact, you have studied this conception, TI, therapeutic index. In antibiotics, we call the therapeutic index is the CI, chemotherapeutic index. That means LD50 to ED50 or LD5 to ED95. This ratio, we call this chemotherapeutic index. Because we have studied the TI, we know the value of CIOTI larger. That means more higher effectiveness and lower toxicity. That means the larger the value is, the great, the more safety can be used in clinic. Okay, so next one, MIC. The full name of MIC is the minimal inhibitory concentration. MIC is the lowest concentration of antimicrobial agents that prevents visible growth in 24 hours incubation. We call this MIC, minimal inhibitory concentration. Compared to MIC, another one is MBC, minimal bactericidal concentration. Very easy. MBC is the lowest concentration of antimicrobial agents that kill visible growth in 24 hours incubation. We call this MBC, minimal bactericidal concentration. The last one is PAE, post-antibiotic effect. PAE shows the antimicrobial effect after the concentration decreased below MIC. PAE. So let's explain what's the meaning of post-antibiotic effect. Means after we use the drug, while we withdraw the drug, and the drug's concentration below MIC, but this drug still can kill or inhibit bacterial growth in the body. So we call this effect is post-antibiotic effect, such as later we will study aminoglycosides, streptomycin, gentamicin, artemisinin. These aminoglycosides have obviously post-antibiotic effect. Means when we withdraw the aminoglycosides and streptomycin, gentamicin still can kill bacteria in the body. We call this post-antibiotic effect. Okay. Just now we have studied some professional terms, and then we will study the total mechanisms of all antibiotics. Total mechanisms. So that means we also divide antibiotics divided into these 5 kinds. The first mechanism is some drugs inhibit bacterial cell wall synthesis. The second mechanism is some antibiotics can inhibit bacterial cytoplasmic membrane synthesis. The third mechanism is some antibiotics can inhibit bacterial DNA and RNA synthesis. The number 4. The fourth mechanism of antimicrobial drugs can inhibit bacterial protein synthesis. The last mechanism is some drugs can inhibit or regulate the energy metabolism of bacteria, inhibit bacterial folic acid synthesis. So let's study the details. The first mechanism is some drugs can inhibit bacterial cell wall synthesis, such as you should know the bacterial cell wall is composed of mucopeptides. The mucopeptides is a linear polymer composed of alternating units of N-acetylglucosamine and N-acetylmuramic acid. And then these polymer strands are cross-linked by one enzyme. This enzyme is transcriptase. This cross-linking is of great importance because it imparts rigidity and determines the shape of the cell. So just now we explained the cell wall synthesis. And during the cell wall synthesis, during this process, one enzyme very important is this enzyme, transpeptidase. So that means if we can use some drugs inhibit this enzyme, inhibit transpeptidase, that means inhibit cell wall synthesis. Because cell wall determine the shape of the bacterial cell. So later we will study penicillins and cephalosporins and vancomycin. These drugs can combine with the inactivated the transpeptase and then inhibit cross-linking, inhibit cell wall synthesis. And this drug can kill bacteria. So the first mechanism, penicillins, cephalosporins, and vancomycin can inhibit transpeptase, inhibit cell wall synthesis, and they are bactericidal, can kill bacteria. Okay. The second mechanism, the second kind of antibacterial agents can inhibit cytoplasmic membrane synthesis. Cytoplasmic membrane is to control the internal composition of the cell by action as a selectively permeable barrier to some substances, such as amino acids, such as nucleotides and inorganic ions. For example, some antibiotics, such as polymyxin, can damage the cytoplasmic membrane. Damage the cytoplasmic membrane. For example, this is the cell wall. And just now we studied penicillin, cephalosporins, and vancomycin inhibit cell wall synthesis, means the water outside of cell bacterial can flow in bacterial, and the bacterial expansion and then kill the bacterial. The second layer is the cytoplasmic membrane. And just now we explained some drugs, such as polymyxin, can damage the cytoplasmic membrane synthesis. So like this membrane be damaged, and the inner bacterial amino acids, nucleotides, and inorganic ions flow out, and the bacteria will be killed. Okay. The third mechanism of antibacterials can inhibit bacterial DNA or and RNA synthesis, such as quinolones. Later we will study quinolones. For example, ciprofloxacin, oxyfloxacin, they are quinolones, can inhibit bacterial garase, inhibit the bacterial DNA synthesis, can kill bacteria. So quinolones are broadly spectrum. They are bactericidal agents. These drugs can inhibit bacterial DNA synthesis. And rifampicin is one kind of broad spectrum antibiotics. And these drugs inhibit bacterial RNA synthesis. And this drug is the broad spectrum bacteriostatic agent, can inhibit bacterial RNA synthesis. In clinic, mainly used to treatment of tuberculosis. Okay, next one. Some drugs can inhibit protein synthesis. For example, some drugs, in fact, most drugs, the antibiotics which interfere with bacterial protein synthesis in bacterial includes these drugs: chloramphenicol, erythromycins, infects, macrolides, lincomycin, clindamycin, and tetracyclines, aminoglycosides, such as streptomycin, kanamycin, and so on. These drugs inhibit protein synthesis. And for example, erythromycins, macrolides, and chromycin. Erythromycin is the narrow-spectrum bacteriostatic agents, inhibit protein synthesis. Similar to erythromycin, lincomycin, and clindamycin, they are same with erythromycins. Tetracyclines and chloramphenicol, they are broad spectrum bacteriostatic agents, inhibit protein synthesis. Aminoglycosides, streptomycin, kanamycin, they are aminoglycosides. They are narrow-spectrum bactericidal agents, inhibit protein synthesis. Okay. The last cats can interfere with bacteriophilic metabolism. For example, later we will study sulfonamides or TMP. These drugs can interfere with folic metabolism, and they are broad spectrum bacteriostatic agents, inhibit folic acid synthesis. Okay. Just now we studied the total mechanism of all antibiotics. And we also can divide them into 5 cats. The first mechanism, so for example, it is the bacterial. The outer layer is the cell wall. And some drugs, such as penicillins, vancomycin, cephalosporins, inhibit cell wall synthesis. And the second layer is the cytoplasmic membrane. Some drugs, such as polymyxin, inhibit damaged cytoplasmic membrane synthesis. And the third mechanism, lots of drugs can inhibit bacterial protein synthesis, such as erythromycins, tetracyclines, and aminoglycosides. And then some drugs can inhibit bacterial DNA synthesis. For example, quinolones, such as rifampicin, inhibit RNA synthesis. At last, the fifth mechanism is modification in energy metabolism. They are sulfonamides and TMP inhibit folic acid synthesis. Okay, so we studied the total mechanisms of all antibiotics. So we also can divide antimicrobial drugs into 5 cats. The first cat, inhibitors of cell wall synthesis, vancomycin and penicillin cephalosporins. The second cat inhibit protein synthesis, such as aminoglycosides, tetracyclines, erythromycins, and chloramphenicol. The third cat inhibitors of folic acid synthesis, folate antagonists, sulfonamides, and TMP, trimethylprime. Number 4 kind of antibacterial drugs. They are inhibitors of nucleic acid function or synthesis, quinolones, all and rifampicin. The last cat, the inhibitors of cytoplasmic membrane, such as polymyxin. This class, we studied some professional terms, and we also divide antimicrobial agents divided into these 5 cats. Next class, we will study different antibiotics. So next class, we will study penicillins and cephalosporins. And then next class, we will study aminoglycosides. And then tetracyclines, erythromycins, their pharmacological effects, clinical using, and adverse reaction. Okay, class is over. See you next time. We will study different antibiotics.